So what happens as we grow older, this whole machinery starts showing decrements, in particular cytochrome oxidase. And the mitochondria as a whole is designed that when the certain level of their metabolic machinery does not operate for the oxygen consumption and the ATP production, it self-destructs.
Welcome, everybody. This is Buck Jarvie with the Longevity Roadmap Show. And today, very fascinating show, Dr. Francisco González-Lima. We are going to talk about what I think it's an incredibly interesting topic, which is brain metabolism and what happens to brain metabolism as we age. In other words, the brain is an incredibly high energy consuming organ. And if you remember from Bio 101, or if you've, maybe you're more advanced as a science person, but the power centers of the cells are the mitochondria. And if you also remember from biology, there's the electron transport chain that creates ATP, which is sort of the central, energy fuel. Dr. Lima's research focuses on that, the mitochondria and the electron transport chain, especially the last part of that chain, which involves an enzyme called Cytochrome Oxidase. And this show gets into the weeds. And so if you really like science, you're going to like it. And you might not understand 100% of it. Honestly, there is, we do get into detail, but I think the big picture is that essentially you've got this paradox of, aging when it comes to mitochondria. Mitochondria inherently, when the mitochondria function the way they're supposed to and the electron transport chain function the way it's supposed to, it releases reactive oxygen species or free radicals around it. And you probably heard that those can actually create tissue damages within the cells, within the DNA of the mitochondria itself. And so it's a machine that's somewhat inefficient in that, the byproducts of it are actually damaging the mitochondria itself. So the dilemma then becomes, well, how do you potentially intervene with that? So we talk a lot about his research in that space, and we focus on a couple of different interventions. One of them with methylene blue, which, I know some people are using methylene blue as a supplement. I do not. I just think one of the reasons that I don't, you will learn about from the show, because the idea here is methylene blue potentially can be helpful at lower doses, but if you don't get that dose right, it could actually be harmful. The other thing that we talk about in the show is the role, I think more interestingly, of light, specifically of near infrared light that is delivered to the brain through the skull, and the potential promise of that. I've heard quite a bit about that, and some very promising things about that in terms of cognition in pretty healthy people who just want to feel sharper and that kind of thing. At any rate, this is a long show, but if you like science, if you are into getting down in the weeds of this kind of stuff, I think you're going to love it. We'll have this show right after these messages.
Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to the show, everyone. Today, my guest on the Longevity Roadmap is Dr. Francisco González-Lima. He's a George I. Sanchez Centennial Professor at the University of Texas at Austin, internationally recognized for his pioneering work on brain energy metabolism, mitochondrial function, and the role of cytochrome oxidase in cognition, memory, and aging. His research has helped shape how we think about neurodegeneration and the science of maintaining brain health across the lifespan. Thank you so much for joining us.
My pleasure.
So, Dr. González-Lima, why don't we start with this? How did you start getting interested in the study of brain energy metabolism and its role in aging?
Well, the first steps happened long ago, about more than 40 years ago, when I realized the relevance of energy for brain function. And in the early days, my interests were more in neuroendocrinology when I was an undergraduate at Tulane University. And that was a wonderful time because I had an experience my last summer there working on an undergraduate honor thesis that with a group that discovered some of these neuropeptides, these hypothalamic hormones. They shared the Nobel Prize only a few months after I left the lab. And this was the beginning of my interest of how neural function and metabolic functions were linked. And when I was then working for my graduate degrees and as a postdoctoral Humboldt Fellow in Germany, I really grew my interest. And in that German group, we were the ones that developed the fluorodeoxyglucose method in animals that was used as the first functional neuroimaging mapping technique that was translated to humans using PET, positron emission tomography. So for the first time, we were able to map metabolic function not only in animal brains in vivo, but also in humans. So that was the beginning of the field of functional neuroimaging. And it's all based on metabolic markers. Nowadays, more so in hemodynamic or blood flow related changes.
You mentioned the importance of brain metabolism when it comes to brain function, obviously very important. So in the big picture, kind of give us a sense for, even in non-disease states, what is happening over time as we age to brain metabolism?
Yes, there are many phenomena that happen during aging, but focusing on the energy metabolism, one of the most prominent changes is what happens with our mitochondria. The mitochondria, these intracellular organelles, they are in a symbiotic relationship within our cells. They are there to provide this energy in the form of adenosine triphosphate or ATP. In exchange, the cells provide just about all of their needs, but they originated as bacteria. So, for example, their DNA is not a double helix, it's a circular DNA. And to maintain the symbiotic relationship, mitochondria only have a very limited amount of DNA on their own that is devoted for the proteins that they need to survive and produce the energy. And then the rest has to come from proteins that are derived from nuclear DNA and shipped into the mitochondria. So the mitochondria is something that we have in every cell. And the more energy demanding a cell is, the more mitochondria we have. What happens as a function of aging is that some of these mitochondria starts accumulating defects. Some of these defects are linked to their so-called cellular respiration in the process of so-called oxidative phosphorylation, especially when you have excess oxygen that is not fully reduced into water. That becomes a reactive oxygen species. And there is a key enzyme there This is the last enzyme in this so-called electron transport chain. That is the one that converts or catalyzes the reaction of oxygen into water. So when water is fully reduced, it's non-reactive, but that reaction is the one that is coupled with the oxidation of adenosine to produce ATP.
And that is a cytochrome oxidase, correct?
Yes, cytochrome oxidase, or sometimes it's called cytochrome C oxidase, or complex 4, the terminal complex of the electron transport, and the rate-limiting enzyme for oxygen consumption. So what happens as we grow older, this whole machinery starts showing decrements, in particular cytochrome oxidase. And the mitochondria as a whole is designed that when the certain level of their metabolic machinery does not operate for the oxygen consumption and the ATP production, it self-destructs. So, for example, cytochrome oxidase or cytochrome C oxidase, the name comes because there is a very small protein that is called an electron carrier, cytochrome C, that passes that electron to cytochrome C oxidase, and which passes to oxygen. Oxygen, being the ultimate electron acceptor, is the one that is pulling all of these electrons through.
Mm-hmm. So in the big picture, just to summarize, so make sure everybody's on the same page, that, basically you've got, the brain is very, very energy, it requires a lot of energy, a lot of consumption of energy. People know in general, the mitochondria, we talk about it as this energy production centers. You learn that from basic biology. And the electron transport chain is a big part of that. Electron transport chain has a unique thing in that it actually creates free radicals, creates reactive oxygen species, which it needs to do because that's kind of what the reaction is. But then in the process ends up damaging itself. And in particular, you're looking at damage to the last step, basically in the reaction, which is through this enzyme cytochrome oxidase. And cytochrome oxidase itself seems to get, am I right? It seems to be damaged more in these as we age.
Yes, that would be the simpler way to put it. Yes, but since it can no longer take up this cytochrome C in an efficient way, then the membrane of the mitochondria that maintains all of these organelles and cellular membranes and mitochondrial membranes, they have permeability to certain compounds or ions. So this permeability changes, and when the efficiency of the electron transport is compromised, and then the cytochrome C, the little electron carrier protein, is able to go out. And this cytochrome C, not the big enzyme of the little electron carrier, becomes a signal for the cell to self-destruct. There is called an apoptotic signal. So the system is designed that the mitochondria that are defective and the cells that harbor more of these will eventually be eliminated. But in order to try to compensate for this, when a mitochondria starts having lots of, for example, mutations in their circular DNA, it starts to try to divide at a faster pace. So as time goes by over the years, then what you end up having is what is referred to as a mosaic. In other words, some mitochondrial lines, the healthier one, you end up having less, and you end up having more of the one that carries some of these point mutations. They're still viable, but they are reproducing at a faster rate because the only cells with a couple of sections that cannot reproduce to renew themselves in our body are these neurons. They carry these defective mitochondria that are piling up and becoming a greater proportion of the overall amount of mitochondria. And this is often referred to as the mitochondrial theory of aging, that is very closely linked to the so-called free radical theory of aging, because the free radical is the reason that the mitochondria are developing these mutations, and the mitochondria failing and generating this disproportionate larger amount of defective mitochondria is what is resulting in this metabolic compromise. So this is really the connection between the mitochondria and this aging-related compromise in the ability to produce energy using oxygen.
So is there a reduction in the brain in it was certainly you've talked about, a reduction in fully functional mitochondria because, because of these mutations, but is there an actual reduction in total number of mitochondria as well?
No, the total number is actually increased.
Okay.
And we know
because
this,
it's trying to overcompensate. Maybe.
Trying to overcome. So, for example, when this happens in muscle, where it's another tissue, there are lots of mitochondria. In mitochondrial disorders that are more pronounced, that this slower change that I mentioned that happens over the years. One of the things you see in this tissue is more mitochondria, but more defective mitochondria. So ironically, they have more mitochondria, but they have less capacity to produce aerobic or oxygen-dependent energy.
The analogy I think of is like in heart failure, you have inefficient pumping and then the heart gets bigger. In that case, it's hypertrophy, not but you're basically trying to make up, you're trying to compensate.
Correct. It's
right,
compensatory phenomenon. So this happens during aging, and it is something that if we could do two things, if we could reduce the amount of these oxygen-free radicals that are the ones that start this cascade of free radicals, but at the same time maintain our levels of energy production, ATP production, this is what becomes then the ideal situation. For example, some people that are doing research, they will tell you, well, caloric restriction will lower the level of this oxidative damage. However, when you do that, yes, you get less oxidative damage, but you get less ATP production, less energy production.
Yeah. The paradox in the whole thing is that we need that. We need it to do what it does, and it creates the very thing that destroys it, or that that causes mutations. So that's, I guess, the therapeutic challenge, or that's the challenge in science, is how do you thread that needle, right?
Well, we have been able to overcome the challenge, because the key for this problem is in cytochrome oxidase itself, because what is producing the oxygen-free radical is when you don't have enough cytochrome oxidase in the active form, the so-called holoenzyme that is able to catalyze the reduction of oxygen to water. That, for example, if you're doing exercise, you accelerate your oxygen consumption, but you may reach a point where you go like,
yeah.
There is enough air and enough oxygen there, but the limiting factor that is not allowing you to produce more ATP is that you already saturated the capacity of the available cytochrome oxidase in your mitochondria. So that's the rate limiting there. So normally when we accelerate the rate of oxygen consumption, then some fraction of oxygen would not be fully reduced, and this is what is called, for example, superoxide. And then there's a cascade to try to add some hydrogens to make it for example, from superoxide to hydrogen peroxide, to hydroxy radicals. It's trying to make it into water, the body with other enzymes. So these are so-called antioxidant enzymes. But in reality, the major and first antioxidant enzyme is cytochrome oxidase itself. So what you need to do is upregulate cytochrome oxidase function to minimize then the production of this reactive oxygen species, because you have more enzymes. So if you increase your breathing rate, your demand for more energy, you will have less of these reactive oxygen species that are being formed.
What are some of the interventions that you're looking at to tackle this issue?
Yes, the first intervention that we used was following a pharmacological model. Try to see, is there any chemical that can intervene and act on this process? And this is what led us to methylene blue, but not just methylene blue in general, but low dose methylene blue, because only the low dose has the capacity, when you administer that to an organism like a human, to reach an equilibrium between the oxidized and the reduced forms of this chemical dye. And when it reaches an equilibrium, it just cycles electrons between the two forms, the oxidized and the reduced. But these electrons he takes from the immediate environment that is surrounding the chemical, and he can also provide those electrons to the surrounding other chemicals. So what is magical about this, and I'm using this term as it was the first way that it was referred to, when methylene blue was first injected in a living animal. It was a living rat, and then the animal was dissected. The surprise was that the blue was concentrated in the nervous system, in the brain and other parts of the nervous system throughout the body. So it has this redox cycling that happens with low concentration, has an affinity to get inside highly respiring cells where there's a high movement of electrons to oxygen. And this, in reality, it not only just gets inside the cell, but it concentrates inside mitochondria, where that electron transport and exchange is happening with the oxygen that we are shipping into this cell respiration. So it is, even though it's administered systemically, it goes and concentrates, has a mitochondrial stain, and which is primarily localized in the nervous system. Of course, it goes in any cell that is engaged in cellular respiration, but it's just the large concentration that will lead to that visible appearance when you look at it. So there you have a chemical that does this. I have not been able to find any other chemical that has this property. It's called auto-oxidizing, but an auto-oxidizing property that at very low concentration reaches this equilibrium.
Obviously, we know it gets to the brain and what the theoretical purpose or benefit is there. I mean, what kind of things have you actually seen in terms of metabolism or, related to the health of an animal?
Well, two things. First, in order to be able to monitor these things, I have to develop a quantitative histochemical technique for looking at the brains of animals. And this I did long ago. It was the first quantitative histochemical technique developed, and it's based on enzyme histochemistry. In other words, the more concentration of this working enzyme that is in the tissue, in the nervous tissue, the more oxygen that can be consumed, and one can tie this process with another chemical that changes its color, the more oxygen consumption that is going on. And then you set this up in a way that there is a linear relationship between the oxygen consumption, the concentration of the active enzyme, and this changes in optical density in the tissue. So this allows us to study these changes in animals and manipulate animals. For example, give them methylene blue at low concentrations, see directly the effect on cytochrome oxidase in the tissue. And then we move from there to humans. The reason I wanted to do this in humans is in healthy people, this should be a benefit to be able to accelerate mitochondrial respiration. But also, I was motivated by one of the dissertation works of one of my students, John Pala, that in which we analyzed fresh frozen brains from Alzheimer's patients. In order to do this, it was a unique study. We couldn't get these fresh postmortem brains from anywhere in the world, including all of these brain banks. It was too late, these postmortem tissues. So we went to a place in Arizona, it's called Sun City, Arizona, where retired people live there. And they have a healthcare system there where they get the donations of this brain and they can quickly freeze them. And we also were there when the pathologist was informed that there was somebody has to certify death. We will be there. We'll remove the skull and take the brain. And my job was to dissect the brain in parts that could be identifiable and labeling everything. And I collaborated then with the scientists there at the Sun City Health Institute. And I will give them a piece for this region, and I will get another piece. And when I have all of these things with my student, we ship everything to Austin. And then we start analyzing. And what we found is what we suspected from the human functional neuroimaging studies, particularly the PET studies, that were telling us that there was this energy deficiency in the brains of these Alzheimer's patients when they were alive. But the major change was a decrease in cytochrome oxidase activity. And we histochemically could demonstrate this directly on the tissue. And this was particularly pronounced in the superficial layers of the cerebral cortex. And these are the layers where we have the largest decrement and atrophy of neurons as we grow old. And these, however, the main problems of these brains were not due and were not correlated with things like the amyloid deposits or the neurofibrillary tangles. So this was a phenomenon that was happening independently of these changes. In other words, it was the opposite situation. The reason the cognition was being impaired was because of an energy metabolic failure. And this was related to mitochondrial compromise. It was not that there was some abnormal protein that was damaging the cells and then was leading to these. So, in other words, we saw this opposite of what is the dogma in the field of Alzheimer's. So from there on,
is the suggestion that somehow, and I've heard this theory in the context of, people talk about type 3 diabetes and all that with brain metabolism of glucose. But is the thought then, is the idea potentially that the decrease in metabolic capacity in these neurons is causal, and that, the other things that we're seeing, the structural changes, may be sort of secondary to the initial
consequential?
Yeah,
you are right. That's exactly And we like to call this the scientific theories. But what I can tell you is that we have seen that in animal experiments where we can test causality, that's exactly what's happening.
In theory, doctor, wouldn't you be able to then induce some of these structural changes in animals by creating problems with cytochrome oxidase?
Oh yes, and this was long ago. It was the first after we did these findings. The first so-called Alzheimer's disease animal model was inhibition of cytochrome oxidase activity using a mitochondrial toxin, and these animals will start developing. You have to do this at a very low level, because if you stop, if you inhibit more, I would say you could go up to a third of the overall mitochondrial cytochrome oxidase activity inhibition. But if you go beyond that, you will die. To give you an example, the most classic metabolic poison in the world is cyanide. What does cyanide do? Cyanide directly inhibits cytochrome oxidase inside the mitochondria. It occupies the binding site where oxygen is normally going to be located in order to be reduced to water. And by producing this so-called allostatic inhibition, you quickly die. So you can only reduce this, but not entirely inhibit this, or like using a genetic approach, mutating the enzyme in such a way that it's not viable. You don't have a viable organism. But you can reduce it pharmacologically, and then demonstrate. The animals start showing first the problems with memory problems. Start out with short-term memory problems, and then the problems move to more recent memory, to more long-term memory.
Do they result in the neurofibrillary tangles and all those?
Well, that's where the thing is different. We don't see any significant amount. In other words, we don't see any causal relationship between cognitive impairment and amyloid and deposits. I would not say neurofibrillary tangles, because neurofibrillary tangles, when they are inside a neuron, that neuron is metabolically dead. In other words, that neuron is a ghost. You may still see it, but inside it has this tau fibrillary proteins that do not allow the neuron to operate, and eventually it would just be removed. But the so-called extracellular concentrations of this amyloid, like the beta amyloid, it is a phenomenon that starts happening as a compensation for this decline in this energy metabolism. And that we see. And in fact,
you see amyloid. You do see amyloid deposits.
You see amyloid, but it happens as a consequence of this, and it doesn't happen at the beginning. It happens later on. For example, if you look at the embryonic brain, in which there is a lot of neurons that are dividing, but there is a lot also of neurons that are being trimmed down, that are dying. So there you see the largest amount of beta amyloid in our brains is when the brains are developing. So we know beta amyloid is a reaction, a consequence to these neuronal losses that are happening. And it's not a part of the causal events that are leading to the dementia. And this is a major difference between the individuals like me and others that are sponsor the so-called vascular hypothesis of dementia. And I have defended, along with them, that these are the most important things that are leading to the common type of dementia that we see in old age, what I refer to as geriatric dementia, which is not Alzheimer's disease, like originally defined by Alois Alzheimer's in a younger patient. It is a confusion that has set back Alzheimer's research for over 50 years.
Going back to your discussion on methylene blue, in this model, you would think that there might be some promise for methylene blue to slow down the advancement of Alzheimer's disease.
Well, only if it's done properly, that is using low dose that leads to this auto-oxidizing cycling equilibrium. The problem is that in vitro, if you use methylene blue, you can, for example, reduce the aggregation of tau in the test tube. And it's a linear function. The more methylene blue you have, the less tau aggregation that happens. That was a finding discovered by British scientists. But then they led to their idea that methylene blue could be an anti-tau aggregation agent. And then they started doing clinical trials with high levels of methylene blue that, yes, they have this anti-tau aggregation property, but at those levels, they are toxic. And they do not have any of these metabolic properties that I told you. That is the properties of being able to donate electrons to the electron transport and to reduce oxidative damage and increase ATP production. So by using methylene blue, but instead of using low-dose methylene blue, they are not achieving. So ironically, then in their clinical trials, they were using very low-dose methylene blue as a control for the ones where they're giving the pharmacological levels that in vitro would produce more reduction in tau aggregation. And the patients who benefit were the ones getting the control low-dose methylene blue, and the ones getting the higher ones were not benefiting. And also there was a confound in some of those trials where methylene blue was not used as a monotherapy, but it was used in combination with so-called Alzheimer's drugs like cholinesterase inhibitors and memantine, which in fact do harm to the brain more than benefit brain function in geriatric dementia patients. So in those studies, it's confound. And what they found in those studies was when methylene blue was used at a low dose and as a monotherapy, then there was a benefit, the cognitive benefit. But if they combine it with these other drugs that are working against energy metabolism and have methylene blue at higher doses that are creating toxicity and are not benefiting energy metabolism, then so this was probably, in my opinion, the worst thing that could have happened to methylene blue.
Yeah, they looked at it maybe through the wrong lens, right? So
yeah, and that's why I always say methylene blue is a hormetic drug. That is, at low concentrations and high concentration, it has opposite effects.
Yeah,
completely opposite. Even for the most common use, that is in the emergency room.
Right, for carbon monoxide poisoning, right? So
yeah, carbon monoxide poisoning, cyanide poisoning, any of these metabolic poisonings that interfere with either oxygen transport or oxygen utilization. If they give a low-dose methylene blue, they can prevent what is called methemoglobinemia, which may be caused by this carbon monoxide, by displacing that from the heme part of the hemoglobin. The hemoglobin carries the oxygen. So if we imagine this is the oxygen, the hemoglobin carries the oxygen, and the other heme protein that receives the oxygen is cytochrome oxidase. It also has the heme group, same carries. So this is what happening through our blood, hemoglobin carries, and then inside the mitochondria, this gets it, and then it converts it to water. These poisons that I'm referring to are affecting that pocket, that heme pocket. So, but if you give a high amount of methylene blue, you induce methemoglobinemia. In other words, there is so much methylene blue that it actually competes with oxygen instead of just displacing it from the heme component of the protein. So this is something that needs to be understood, and that group that led to these studies and created a company around it never understood this, even though they had all the scientific research available to them. And not only that, I think one of the consultants was one of our former trainees who explained this to them and who told them, you cannot do this. And they did some monkey experiments where they were using these larger doses and larger doses. She explained to them, this is just going to be toxic and damage the monkeys and kill them. Sure enough, it was a disaster. So what they did is that they used as high as they could get away with the toxicity, thinking always that the more, the better. Or just like I told you with methemoglobinemia, no, the more is not the better. If you get more, instead of saving somebody, you are promoting methemoglobinemia, even in acute conditions.
Another type of intervention that you've looked at is photobiomodulation. Tell everybody what that is and exactly what how it works. And
the type of photobiomodulation that I started investigating thanks to one of my trainees, Julio Rojas. This is a dissertation. He's an MD, PhD, now professor of neurology, UC San Francisco. And he was the one who indicated to me, look, one of the leading researchers in cytochrome oxidase, Dr. Margaret Warren Riley, has done this in vitro study with retinal cells, ganglion cells in the retina. And she shows that if you have mitochondrial toxins, light in the red to near infrared is able to antagonize this poisoning. And we got interested into these and did some in vitro work, but then we used the eye in a living animal. We did mice and rats, pigmented eyes in a living animal as our first testing ground, creating a retinal degeneration that simulates the most common type of blindness in young people. It's called Leber's optic neuropathy. So we knew ganglion cells will die in response to this mitochondrial toxin. And then we injected methylene blue in some experiments, and in others we used the photobiomodulation in the eyes. And what we found in both cases is that we could prevent this neurodegeneration. And so once we saw this, and I wanted to do it in the eye first, because that way I knew that light was going to get through to the back of the eye, to the retina. And then we started experimenting, can this go through transcranially? And sure enough, in animals, even with the red light, you could have it transcranially. And we could demonstrate this. We could take the brains out. We could measure cytochrome oxidase activity. We could measure oxygen consumption. We could put a probe inside the frontal cortex and in vivo measure the changing oxygen consumption. We were convinced and test the animals. For example, if it is a visual function, you can test visual discrimination. You can see that the animal can improve or you can prevent the visual deficit. You can see the changes at the molecular level, and you can see the histopathology. You can see the loss of the neurons when you don't have it, and essentially you can prevent that So that encouraged us to go transcranial. And then we published this for the first time in the journal Neuroscience in 2008.
Can I ask, what is the mechanism that would be working there? We've been talking about the effects, of problems with cytochrome oxidase.
Cytochrome oxidase. Yes. Well, that's the key. Cytochrome oxidase, like the name implies, cyto means cell, chrome color. Cytochrome oxidase, this large enzymatic complex, is the main photon acceptor inside cells. In other words, it's what gives cells the color. It absorbs the light at certain wavelengths, and it reflects the other wavelengths. So it absorbs light primarily in this red to near infrared range. And when it does that, those photons are absorbing. They operate in the sense of the electron transport in the same way that adding an electron to the enzyme was working. The photon is just like an electron, but an electron has negligible mass, but the photon has no mass. So when the photon is absorbed by cytochrome oxidase, the enzyme emits an electron, and that is taken by oxygen. Oxygen is reduced, oxidative phosphorylation continues, ATP production continues. So it is a way to, instead of donating electrons like methylene blue, you are donating photons that when absorbed by the same enzyme, then if they're absorbed by some other, photons can be absorbed by any chemical, but those chemicals are not coupled with this process of reducing oxygen to water that is coupled with oxidative phosphorylation and ATP production. So that's what makes it important. And the fact that that enzyme is the major photon acceptor inside the cell. So that's what we not only study this phenomenon in animals, we were able to measure this for the first time in humans, non-invasively. We had developed a technology to do this, and we can monitor, we can have sensors here and monitor the levels of this. I refer to this process as photooxidation of cytochrome oxidase.
How can you tell, what's going on? And are you using some sort of imaging to show what's happening in terms of the effects of the light transcranially?
We use a type of optical imaging. You likely have heard about the functional near-infrared spectroscopy. That you can measure oxygenated hemoglobin versus deoxygenated, just like I was telling you in the example. So the oxygenated hemoglobin, the oxygen is trapped. When it releases the oxygen, it changes conformation. So the same hemoglobin, depending on whether it's oxidized or oxygenated or deoxygenated, it changes colors. So that what gives you the blue and the red blood, the deoxygenated and the oxygenated blood. So this is the same technology that you use for oxygen saturation in your finger. But over there, you are looking at one or at most two wavelengths that are absorbed mainly by oxy or deoxyhemoglobin. But in order for this to work for cytochrome oxidase, we created what is called broadband near-infrared spectroscopy. In other words, you give 1,000 different wavelengths. And with all of these wavelengths, you create the signature, because every chemical has a signature of what different photon wavelengths it absorbs. So by doing this, one can determine the concentrations of cytochrome oxidase and how these concentrations are changing in response to the absorption of photons.
What do we know from any kind of human trials right now in terms of photobiomodulation, in terms of potentially improving cognition in elderly or even neurodegeneration?
Sure. Our first switching from the animal to the human, the first report of our study was in 2013 in Neuroscience, the flagship journal of the International Brain Research Organization. And there we show for the first time in a control study, sham control, placebo control, blinded, that shining infrared light that can penetrate through the forehead, and we use an optimal wavelength from our studies.
What would that be? What's that wavelength?
1064 nanometer, 1064.
So you mean there are devices you can get the 1064 wavelength. So yeah.
And that's what's one of the reasons we use it, because those lasers have been clear as safe for human use for other applications. And we use them in a very low power levels. To give you an example, for applications, non-destructive, non-ablative laser applications, but we use only like 3.5 watts in those devices. Instead of for other applications, you can use like 20 watts. And so the overall power density that we deliver through the forehead in humans is 1/4 of a watt per centimeter square. And this is a very low intensity, but you don't need more than this, because what you do is that you just continue with the exposure to those photons with more time. So in terms of optics, dosing is somewhat different than pharmacology. So energy equals the power, the intensity, multiplied by the exposure time, like watts by seconds. It gives you energy that is measured in joules. So our dosing is the energy dose, and we achieve that by irradiating for 8-10 minutes. And that gives enough energy dose that you move this. Remember about the hormetic dose response that happened in methylene blue? The same thing happens with photobiomodulation. If you give photons to the system, to this cytochrome oxidase and the electron transport, you accelerate the system. You keep moving those electrons to oxygen, more oxygen consumption, more energy production. But you can overwrite the capacity of the system and then again lead to oxidative stress.
So you can have too much light as well?
Yes, but in practice, in an animal or in vitro, you can demonstrate these hormetic biphasic dose responses. But in practice, in the human brain, through the forehead, you cannot reach.
Yeah, it's hard to get you too much. Yeah.
You cannot get it there because the skin starts absorbing the light and it starts heating up. And the maximum permissible exposure is something like 1 watt per centimeter square. So we remain into the 1/4 of a watt. But people would not stand being irradiated with a level of light, infrared light, that will actually be counterproductive for this cytochrome oxidase energy production mechanism. So it is safe inherently in that way.
How about clinical outcomes? Or what have you seen in actual,
Yes. So at the beginning, we did only healthy young people.
Yeah.
And we did years after years. So we started testing sustained attention, working memory, category learning, executive function. Each one of these is a dissertation from one of my students. Also combine that with aerobic exercise that you can use to improve blood flow, oxygenated blood. So how does it compare with it? We can do better. 8 minutes, somebody sitting, getting this on the forehead, than somebody working out for 20 minutes to their 80 to 90% of their maximal heart rate for their so-called VO max, maximal volume of oxygen consumption. So then I started to look for clinical populations. Which clinical populations? I wanted to look for clinical population where we know there is a hypometabolism in the prefrontal cortex. So because that's where we're targeting. And by the way, we're targeting these, not what other people are doing, that are using through the hair, because the hair with the melanin absorbs these photons and just hits the hair. And then the dosing is completely equivocal. You don't know what's going in. But that doesn't happen here. And the melanin that we have in the skin, even if we have very pigmented skin, it doesn't make much of a difference because we all, in the deepest layer of the skin, we have these melanocytes that are fully loaded with melanin. So those are the main barrier, and everybody has those. And the wavelength that we use is the one where we can get the lowest melanin absorption. If this is the absorption of melanin, other wavelengths, it starts up, and then we keep continuing increasing the wavelength until we find the hit. But before we produce absorption into water that hits tissue. So we use a narrow optical window where there is the least absorption by the melanin, so it can penetrate to your cortical tissue, but it will not hit the tissue either. It is not necessarily the optimal one for the cytochrome oxidase enzyme in terms of its peak of absorption. But if the photons do not get to the nervous tissue, it doesn't matter if they are in the absorption peak of the enzyme. So it is a compromise that we use. And we use the 1064 because it was safe to use in people, but it's around that 1,000.
So were you able to show any benefits to patients with cognitive decline?
Yes. First population that we tried, well, we showed two kinds of benefits. One, if you have a patient, for example, that have depressive symptoms, and you apply to that patient what are called, with my colleagues here in clinical psychology, cognitive therapy. Well, for that cognitive therapy to be beneficial, you have to have prefrontal cognitive function optimized. So when we combine that cognitive therapy with the photobiomodulation, you can then benefit and reduce the symptoms of depression. So in my opinion, that's the best way to use this in clinical populations, because the main effect of facilitating prefrontal function will always be cognitive. However, it also has emotional regulation top down with deeper subcortical areas. And so, for example, with anxiety populations, we tried a whole range of anxiety disorders. We refer to a pathological fear. And alone is enough to produce as a monotherapy to reduce the anxiety by acting on this emotional regulation. In other words, facilitating that these prefrontal areas do the top-down regulation.
How about actual cognitive declines, though? I mean, outside of psychiatric.
Yes, we tried also bipolar patients. We demonstrated that we could improve cognitive functions there, especially executive functions. A particular one that is called cognitive flexibility. It's improved. We have published all of this. And the latest one we did for these bipolar patients, we use people who they are stabilized from the point of view of their mood. In other words, they are remitted patients. They are monitored every week. They to make sure that this is the case. And then we do a weekly treatment, and we improve their cognitive performance in a whole battery neuropsychological tests.
How about Alzheimer's? Have there been any studies in this yet?
Not my group. What we have done is older people with subjective memory complaint.
Sure.
And in 5 weeks, 5 weekly treatments of 8 minutes each is enough to see
Daily? 8 minutes
No,
daily?
no. We did only once a week.
Once a week. 8 minutes. Okay.
8 minutes once a week. We could see, and we had a range of people all the way to 90 years old. We also measure carotid ultrasound, ways to try to look at other phenomena, did some fMRI. It is possible to do daily, but I wouldn't recommend with the laser.
Mm-hmm. This device that you're using, is this something commercially available, Dr. Lima, or is this still?
Yes. The first devices that we used after we did all the animal experiments, the initial devices we made ourselves. Especially my older son, my 2 sons are engineers. They helped me build this device for the lab, but then we use a company that was based in Dallas. The 2 people that ran it, one was an MD, and his brother was an engineer, retired from was a nuclear submarine engineer. They developed this laser. They went out of business, so those were the initial ones that I used. But now one of the former trainees that I was his co-advisor, who got his second PhD in electrical and computer engineering from our university, very talented student, as you can tell. He created his own company here in Austin. And these devices that we use, that he makes, are tailored to what we found in our experiments to work best.
What's the company, just for people who are curious?
I think it's called Cytonsys, like CYTONSYS. I am not involved in his company. He's a former trainee.
No, it's fine. Just probably some people at this point are like, boy, maybe I'll give this a try.
Well, they have developed a whole line. They have like a 3D printed thing that goes into the head, but I really just recommend aiming the forehead. Anything else is questionable. There is a group in Toronto that does it in other parts. They do not use the same. The wavelengths that we use is the only one for which there is experimental evidence that it actually upregulates cytochrome oxidase transcranially in vivo. None of the others had been demonstrated to do that. And in fact, in the particular wavelengths that they use, not in my group, but 2 different groups, one of them, Chinese group, had, for example, tried to test it in working memory, comparing in the same populations the 2 wavelengths, and only the 1064 produced the benefits, but the one in the 800s didn't produce the benefit. And we know from the simulation, so-called Monte Carlo simulations, where we can simulate the photon penetration, that this longer wavelength can penetrate and insufficient amount of photons to be a sufficient energy dose to make a difference, as compared to shorter wavelengths, which are the most popular that you can find out there because it's cheaper to make those other devices. And of course, they can work in other parts of the body, like the skin. In the skin, you can do the effects even with red light that is in the 600s.
Right. Fascinating stuff. Yeah, I mean, I guess the thing that comes to mind for me, one last point, and we've been going for a while. I don't want to keep you for long, but the mitochondria, from what I understand, this is something I I learned interviewing somebody else, but they migrate. They can migrate from one cell to another. Is that true?
Uh, it is controversial. There is some evidence for it, but mitochondria are very dynamic. They're all the time. That's why we refer to them in plural instead of mitochondrial. Yeah, we use mitochondria because they are fusing with each other and dividing and forming these little chains all the time. So there is some evidence. I wish it would be better validated, but there is some evidence that they can move from one cell to another, that they can even go into the circulation. But some of this evidence happens in environments that may not be our in vivo environment.
The reason I was asking is, some people, I've heard of the concept of, mitochondrial transfusions, like healthy mitochondrial infusions via like maybe using platelets or something like that. And I'm just curious on your thoughts on that, because if you can bring new mitochondria into the circulation and potentially get these mitochondria into where we need it, like, new mitochondria, they're healthy. Is that something that you've thought about? Yeah.
Right now, I haven't seen the evidence that would make me feel comfortable. But you're right. Platelets can help transport mitochondria, and it is. But they have other roles. So you have too many platelets is a problem.
Yeah, no, that's a problem too. Sure. Yeah, of course.
And especially when you mention aging, one of the limitations there is your vascular cerebral blood flow. We can improve this mitochondrial function, and in fact, in the older humans that we have tested, the change in the photooxidation of this cytochrome oxidase is larger than in a healthy young person. It's just that we start out with a lower baseline, and we have more room for improvement. And the young healthy person is closer to ceiling. You cannot improve that much. However, there is a limitation in oxygen delivery, because you can have better mitochondrial function, but if the delivery is compromised, this is not going to be the full story. So one of the things what we have to add to the mix is improving perfusion to the brain, and because you need that oxygen in order for the mitochondria to provide the energy.
Doctor, it's been really fascinating talking to you. Thank you for all your time today. Really interesting conversation. I think people are going to learn a lot. And good luck to you on your research. And thank you again for being on the show.
Thank you very much. My pleasure.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.